Hi everyone, today I'm going to be talking about synaptic transmission and then also the blocking of synaptic transmission, which is part of 6.5 neurons and synapses. And this needs to be learned by both SL and HL/IB biology. It is important that before you learn synaptic transmission, you understand what an action potential is, depolarization, repolarization, what voltage-gated ion channels are, and also, this one particularly, how the propagation of impulses in the nervous system works, as it just makes it a lot easier to understand, because I'm also not going to be explaining what that is, but I am going to be referring to it. So it's good if you have that knowledge beforehand. And it's also part of the unit, so you should definitely learn that as well. So, synaptic transmission. So, synapses are junctions between neurons, neuron and neuron, or between neurons and receptors, or neurons and effector cells. And effector cells, an example would be a muscle. Synaptic transmission involves a junction where you have a presynaptic membrane, a synaptic cleft, and a postsynaptic membrane. So in the example that I will be talking about today, I'm going to be using a neuromuscular junction, which in the name involves a neuron and a muscle, where the neuron is here, so the presynaptic membrane, and the muscle is here, postsynaptic membrane. So first, because this is an effector cell, the muscle, you need a motor neuron, so this will be a motor neuron. So the first thing that happens is calcium 2+, so calcium ion channels open. This is an example of a voltage-gated ion channel. So learn about that before you learn about this. As you can see in the diagram, these pink things are the calcium voltage-gated channels. And these channels open, and due to a concentration gradient, calcium will move in. So the calcium ion, Ca2+, will move into the axon terminal, which is step 2. Ca2+ diffuses into the axon terminal. The Ca2, so calcium ions, will then bind to vesicles located inside the axon terminal. As you can see, these are the vesicles, just the green things. And the vesicles contain a neurotransmitter inside them. In the example today, it will be acetylcholine, which is also known as ACH, and those are the little green dots inside each vesicle. Because calcium ions have binded to the vesicles, the vesicles will then migrate to the presynaptic membrane of the axon terminal, and the neurotransmitters will be released into the cleft by exocytosis, where the vesicles will fuse with the membrane and will create like a little gap inside. Actually, I can draw this. Will fuse like this, and then it creates a gap, allowing the neurotransmitters to leave. So acetylcholine is released into the synaptic cleft, which is the gap between either a neuron or neuron. It's just essentially the gap between the two different sides. So in this case, the gap between the motor neuron and the gap between the muscle or the effector cell. Next, the neurotransmitters will bind to receptors, which are these things on the postsynaptic membrane. This is where you should know how impulses are propagated in the nervous system, and this is where it comes into play, as the receptors are sodium voltage-gated channels and will eventually lead to propagation of impulses. So the sodium channels will open, and because they're open now, sodium, which was previously in the synaptic cleft, will diffuse into the postsynaptic membrane, which triggers an action potential. The enzyme acetylcholine esterase, so acetylcholine with an esterase on it, you should know that anything with an ASE at the end is most likely going to be an enzyme. For example, lactose is the substrate, and then lactase is the enzyme. So the enzyme acetylcholine esterase will break down the acetylcholine, which was left in the synaptic cleft when it was released by exocytosis from the axon terminal. And that's essentially synaptic transmission. As you can see here, the direction of the impulse moves from the top to down in terms of the way that I drew it. It moves from the presynaptic membrane into the synaptic cleft and then into the postsynaptic membrane, and that triggers an action potential, which then will move from the postsynaptic membrane here all the way to the presynaptic membrane on the other side of... Actually, no, that won't happen because this is a muscle. If this was a neuron, then it would move to the presynaptic membrane and would continue. So next is the blocking of synaptic transmission. So the example that you need to learn involves neonicotinoids, which are man-made chemicals used in pesticides, so you know that this is going to involve insects. So neonicotinoids, as you can see here in the diagram, are these orange dots. They bind to the receptors in the receiving neuron in cholinergic synapses, and that is a synapse that uses acetylcholine as the neurotransmitter. Because in the body there are obviously a lot of other neurotransmitters, and these include adrenaline, also known as epinephrine, or serotonin or dopamine, and so those can also travel between neurons. So it's not just acetylcholine which is used, it's just an example that is easier to learn because it's also used in both synaptic transmission and then blocking of synaptic transmission. So neonicotinoids will bind to the receptors in cholinergic synapses, and ACH esterase, which is the enzyme that I previously mentioned, will not break down these neonicotinoids, preventing acetylcholine from the transmitting neuron from binding with the receiving neuron. As the receptors are now blocked by the neonicotinoids and acetylcholine can no longer bind to the receptors here, in this case, the sodium channels will not be opened, and so sodium ions can't diffuse in, and therefore an action potential will not be triggered. So this prevents synaptic transmission and causes it to be blocked as an impulse isn't propagated and there's no action potential created in the receiving neuron. This only occurs in insects, and this will lead to the death and paralysis of these insects, which is why pesticides are used in agriculture, as it harms insects. However, it has no effect really on human health. Yeah, that's about it. Okay.